Okay, students, today we will continue our journey through chapter D13. This video will have a short section for standard level, and then it will continue with higher level, but I'll make you aware when the switch happens. Okay, so this is where we left off our previous class, talking about the consequences of mutations in either germ cells or somatic cells. So multicellular organisms like us, most of our cells are what we call somatic cells. They build up our tissues and organs, and these cells can then divide to form other types of somatic cells. So these are the ones you find all over your body. Then we have a special type of organ called the gonads. Those are your ovaries and testes. Here, there's a special group of cells called germ cells, and these produce our gametes, the eggs and the sperm. Now, mutations then in germ cells will produce gametes carrying that mutation, and that will, of course, affect all cells in any potential offspring, because the gametes fuse and they have one giving us a full set of chromosomes, and then that single cell gives rise to all other cells in that future organism. So a mutation in one of the gametes will be present in all cells of that organism. So these are where heritable effects come into place. Heritable traits or heritable mutations are only those that happen in germ cells, or potentially when a germ cell gives rise to a gamete, potentially, I guess. So those are the heritable changes. Mutations that happen in somatic cells have local effects, and they are often, or at least cancers may often be a result of mutations in somatic cells. So that's what we should know about these consequences. Mutation is the main source of genetic variation, and most mutations that occur are either harmful for the individual or neutral. But there is a small minority of mutations that give rise to beneficial effects for the individual in their specific environment. And for a population of organisms, the mutations are the ultimate source of variation that underline evolutionary change. So evolution requires that between individuals in the population there is variation of traits, and those variation of traits ultimately rely on mutations. So this is how we get different types of genes, and new genes can form via mutations and so on. So this is very, very important. So we should definitely keep track of this as the main source of genetic variation. And this is the only little part that was for the standard level, students in standard level. So take your time to take those notes, but then after that, your standard level students can take a break. And now we will continue with the higher level content for D13, and then I will continue with D22 and try to see if we can finish that chapter. Okay, so the first thing we're going to talk about in higher level is that we can kind of cause mutations and use that to study the effects of a gene in an organism. Now, we've been able to edit genomes of organisms for decades with various techniques, from the very early techniques involved radiation, giving rise to increasing the rate of spontaneous generation mutations, but then being able to produce DNA and introduce those to other organisms, either via injecting zygotes or using various viruses to load the virus with a specific genetic trait, and then putting that into other organisms. So allowing the virus to kind of carry the genetic trait that we produced into another organism. Now we have way more advanced techniques, which we'll bring up on the next slide. But one example that the syllabus brings up here is what we call gene knockout. So basically, this is a type of genetic modification to an organism where a gene is made inoperable or knocked out. So typically, the gene or an organism that carries the original version of the gene is called a wild type or WT, whereas an organism that carries a knockout version is called a knockout. The way we do this is either now. There are many techniques, but two examples would involve some kind of RNA interference. So we bring in molecules that interfere with the mRNA for that gene, and that's introduced into the organism. And now, more recently, we also use CRISPR-Cas9 to do this, so force mutation in specific locations of the genome of an organism. But we'll talk more about CRISPR-Cas9 on the next couple of slides, just to show that there are different ways of doing this. Now, if we compare then the knockout mutant to the wild type, we can investigate the function of that gene. So if we knock out the gene and that we hypothesize have some kind of function, we can determine if indeed there is a difference between the knockout and the wild type, and that knockout should be the cause for this variation. So the example that you see on the right would be this camouflage pattern that can be observed in squid, but you can then knock the genes for this type of behavior or mechanism out, and then you get a non-camouflaged version. We use knockout on many types of model organisms, the main one being mice, and we can knock out genes that we think are important for cancer. And indeed, if we knock out some of these genes that regulate cell cycle, for example, we've understood that mutations in specific genes can be the cause of cancer, and this allows us to use as a tool to diagnose if some cells are actually cancerous. Now we will talk about the CRISPR-Cas9 system. So this system has revolutionized the way we can edit the genome of cells. So you do not need to know the basic kind of or this background. So if you don't want to, you don't have to write the following parts that I write on this slide, but the next one will be important. But basically, this CRISPR-Cas9 system has two parts. It is the Cas9 enzyme is a nuclease. It cuts nucleic acids. And we found this system in prokaryotes, I forgot to say. And the CRISPR part, let's see if I can remember this, is clustered regions of interspersed. No. Palindromic repeats is the last part, at least. Interspersed, I think, is the middle part. But anyways, this is basically a library of virus nucleic acids that prokaryotes store in their own DNA. So basically, if a prokaryote bacteria is exposed to some virus that wants to kill it, that this system can cut these into pieces. And they store then this DNA from the virus in their own DNA, and the Cas9 system has this little guide RNA in it. Let's see if my cursor can appear, and I think you should see this part, but maybe not. I hope so that you see this highlight. But anyways, this green part here is the guide RNA. So every Cas9 is loaded with one of these guide RNAs, and the guide RNA can bind complementary to some nucleic acid. And if the guide RNA does, the Cas9 will cut. Now, if a bacteria is exposed to a virus DNA, it will have, if it has been exposed to that virus before, it will have the DNA of that virus in its CRISPR library. it will then also have Cas9 enzymes loaded with the guide RNA for that virus DNA, and the Cas9 will be able to break down this virus DNA before it leads to infection and destruction of the bacteria. So it's basically a defense system allowing it to cut specific sequences of DNA. Now, what we've understood now is, or the way we use this is that we can now modify the Cas9 protein enzyme to carry any guide RNA that we want. So this allows us to give it a guide the RNA to cut at specific places we want on any type of organism. So we can introduce the Cas9 into any type of cell and give it the correct guide RNA, and it will go in and cut the DNA of that organism's DNA, or the cell's DNA. Really cool. So the next part, or the next page will be important. What allows us to do mutations with Cas9 at specific locations. This system then has already been used to treat or do many types of important changes, but it was basically published in 2015 or 16, I think, and was 2 years ago, I think, was given the Nobel Prize in chemistry, if I'm not mistaken. There are many applications, but we only need to know one example. I'm going to give you one here, or two, and you can choose which one you feel is most appropriate. So sickle cell anemia, remember this is a single base substitution that causes this change to hemoglobin protein. And bone marrow cells are the ones that produce the red blood cells. So we've used the Cas9 system, CRISPR-Cas9 system, to change bone marrow cells from patients so that we reverse that mutation. So basically, you can also so when you have a cut in the DNA, you have repair enzymes to try to fix that DNA, and it uses the complementaries, so the homologous chromosome, to kind of as a way to understand how it should put the pieces together. Now you can fool this by giving it other types of DNA, and then it will try to, and it can then include new bases that wasn't there before. So you can basically fool this reparation. So you can both remove a thing or replace a thing and add things to DNA. So it's really a very versatile system, and it becomes targeted compared to other types of gene editing. So basically, this allowed us then to change the bone marrow cells here that produces the red blood cells, so they don't no longer carry the mutation. Of course, to do this, you never have any sickle cell anemia type cells, would require you to replace all bone marrow cells. Usually we try to get this Cas9 system into cells using viruses, and they won't necessarily infect all cells and transfer the Cas9 system to all cells. So you'll have usually a patchwork of cells that are fixed and some that still carry the mutation. But they will still have an improvement of life quality for the patient. HPV is another application you can use. Basically, HPV is a virus that a lot of people have. It's a very common virus. There's a vaccine for it now, of course, that I hope you've all taken. If you haven't, take it. It can give rise to different kinds of things, but there's specific strains of HPV that cause cancer. 99% of all cervical cancers are caused by HPV. And the guys listening today, this will be like, hey, I don't have a cervix, so it doesn't matter to me. Oh, you can get throat cancer instead. So please get the HPV vaccine. And it stops you from getting condyloma, which is these genital warts. So please go. And you can also protect people that you have sex with. So that's also cool. And they may have cervixes. Cervixes? I don't know. Anyways, the next part here we'll skip and bring back to evolution instead, because I think that makes more sense. So that's the end of the 1.3 higher level as well. And now we will go to the 2.2 for another 20 minutes. And then that will be the end of this class. 15 minutes actually, we'll go. Hope you're having a good day otherwise. Now, this is kind of where we stopped in D2.2 higher level, talking about epigenetic inheritance. So we said that epigenesis was this process of going from an undifferentiated embryo or zygote and becoming an organism, an individual, and that requires differentiation of cells and um, adding epigenetic tags throughout your genome to get the specific types of cells with the correct types of proteomes to give the correct types of tissues and all over your cell. So when you then are going to produce gametes again that are undifferentiated and don't have epigenetic tags, you need to remove those. So, however, when this happens, sometimes there are some epigenetic markers that remain, and we get what we call imprinted genes. So this is basically what we should write down, that we have inheritance of epigenetic tags between somatic cells. That's important for your development and differentiation of cells. It's part of the epigenesis. But normally when we produce gametes, we remove epigenetic tags. But in some cases, we have some remaining epigenetic tags in the produced gametes, and those are called imprinted genes. And these can cause heritable changes to gene expression patterns of the offspring. So between individuals, we can get this epigenetic inheritance, and that's what it's called, epigenetic inheritance. So epigenetic inheritance can then affect the phenotype of the offspring, and there's been some studies suggesting that these kind of epigenetic inheritance can last for a few generations. So if a parent is exposed to some kind of environment that leads to a specific pattern of epigenetic tags, that effect can be inherited for several generations. And probably diminish maybe in some case between generations. But if you then continuously are exposed to the same type of environment, this of course will be reinforced every time. How important this inheritance is for evolution is a cause for debate among biologists at the moment, and I'm super happy to talk about it if you want to, about the extended evolutionary synthesis may potentially replacing the modern synthesis of evolutionary theory, but maybe that's for another time. There's one example that we are going to look at for this, and that's in the case of the hybrids Tigons and Ligers. So Ligers are very, very big, and the cause or the current hypothesis is that this is caused by epigenetic inheritance. So remember, Tigons, male tiger, female lion. Liger is male lion, female tiger. Lions and tigers are different from each other. Lions are social, so females may mate with many males or several males and produce a litter that has many different males. Produced by many different males' sperms, whereas tigers instead are solitary. They usually mate once and they produce one litter afterwards. And there's an important size determining gene in lions, or at least probably many, but one that we know seems to be involved in promoting the large size of males. So we have a much bigger difference in size between males and females in lions than we do in tigers. So this gene then is basically in the unmethylated working form for males, because so the males are in competition with each other for producing offspring, and so basically the idea is that the males want to produce offspring that are as large as possible, and therefore get offspring that are large and will be able to outcompete their cub mates that may be unrelated. Whereas the females are instead not interested in that. They instead have a methylated version of this, so that all their offspring are encouraged to have the same size because she's equally related to all of them. So this is what we call a sexual conflict in evolution. So the combined effect of in lions is that you get mid-sized cubs with variation, of course. So you both have the big size and the defense against big size. Now I left out. There we go. So the last part here ended with an empty sentence. So here we go for that. So tigers instead have a gene that code or genes that code for their mid-size, even though they're large, of course, but there's no epigenetic inheritance here that is important. So a tigon then gets the defense against, I want to say defense against black hearts, of course, defense against growing big, and the mid-size gene from the male tiger. So their offspring is mid-sized. Whereas when a lion male and a lion tiger female gets an offspring, you get the unmethylated active version of grow big, and you have the normal grow mid-size genes, and they together make a very large size in the offspring. So that's the idea behind how we get these very large ligers. Okay. And then we continue talking about gene expression with the regulation of using transcription factors. So transcription factors, remember, we talked about these before, but these are proteins that bind to DNA and regulate transcription. So they are important. And there are various sites of DNA called regulatory regions where transcription factors can bind. The promoter is a regulatory region, but there are also other types of sites, for example, enhancer regions, that are outside of the gene and silencer regions. The silencer regions aren't brought up in the syllabus, but this image I found has them, so we'll just briefly mention them. So transcription factors are important at the promoter because without them the enzyme RNA polymerase will not bind, but the transcription factors can then affect transcription by binding to these other sites as well. So any individual transcription factor can be categorized according to their action. So repressors are those that decrease transcription, and they typically do this by binding to the silencer sites, whereas activators instead increase transcription by binding to enhancer sites. So that's how those work. And one way that that you can get a transcription factor that affects gene expression is by hormones. Many hormones produce their effects by altering gene expression or by activating or deactivating proteins in the cytoplasm. Steroid hormones can cross the cell membrane because they're also made of lipid. And in the cytoplasm, there are receptor proteins that when they bind with the steroid hormone, they form a transcription factor complex that then go into the nucleus and affect gene expression. One example we can take of this is testosterone. Testosterone is a steroid hormone produced in the testes, and it travels throughout the body with the blood, and it will basically engage with all cells, but only those cells that actually produce the receptor will form the transcription factor and get the effect on gene expression. And these activated genes, they produce, for example, male sexual characteristics during development and during puberty and so on. Peptide hormones instead, they bind to the surface receptors that then change cellular activity via second messengers, but we don't need that. Another example of how external factors can impact gene expression is looking at prokaryotes instead. I think I mentioned this potentially also for humans, so this example that I bring up is actually only for prokaryotes. I'm not sure if the same thing happens for eukaryotes. But anyways, in prokaryotes, there are basically regions of gene around genes that are called operons. So they include the gene plus closely related regulatory regions. So the operon is those regulatory regions close to the gene and the gene itself. For example, it will be the promoter, the operator, the gene, and the terminator. Those 4 things together make up one operon. Now, the famous example that we're going to use is the lac operon in E. coli. So the gene in this operon codes for the enzyme lactase that is used for lactose digestion. You only need this, so the bacteria, of course, only need this enzyme when there is lactose present. And normally, the operator, a part of that is a regulatory part that's just ahead of the gene, carries a repressor that stops the RNA polymerase. There will be no expression. However, when lactose is present, it acts as what we call an inducer. So it can bind to an allosteric site of the repressor that changes the active site, and it no longer binds the operator. And now there's no blocking of the RNA polymerase, and the lactase gene can be described, and we can start breaking, or the bacteria can start breaking down lactose. Isn't that neat? And the final thing we will mention as a way of gene regulation is that you can also control gene regulation by breaking down mRNA. So there are nucleases, there are enzymes that digest nucleic acids. You have those also for when you're eating. You have nucleases in your gut that break down DNA, but you also have them inside cells. mRNA, of course, is vital for gene or protein production. So the longer mRNA is found in the cell, the more of the protein will be produced, or the polypeptide will be produced. Typically, mRNA may exist for minutes to days in humans, and it avoids breakdown by this poly-A tail, for example. Remember these post-transcriptional modification. So these nucleases don't break down mRNA until this part is reduced. And for some reason, this poly-A tail will start being broken down when the mRNA is no longer necessary. I assume this, I'm not sure how this would be, how the mechanism for knowing when the poly-A tail should be broken down. But when this starts shortening and disappearing, presumably maybe it happens in a regular time span, and then you can maybe make this longer or something if you need the protein or more. Anyways, when the poly-A tail is basically gone, the nucleases get access and they can start breaking down the mRNA. So this process regulates the transcriptome and the proteome of the cell. And that's basically it. The BioNinja also brings up the proteasomes again, that breaks down the proteins, and we talked about these already before. So you can just write that the breakdown of proteins by proteasomes using the ubiquitin mechanism. I've already mentioned this. May also be an important way of regulating the proteomes of cells, but I don't think we should mention this more again. And that, my friends, is it. So this whole video, it's 30 minutes, but I know I talk fast, so hopefully this corresponds to 1 hour of lesson time. Let me know if there's anything you would like me to repeat in class, and then we shall do so. Otherwise, have a nice day. See ya.